Hello, welcome to another edition of the CNA's Opus. My name is Rich. I'm a certified nursing assistant. I'm sorry I don't have a lot of time. I gotta go to work, and so I'm gonna through this really quickly. It's just to make a point. This here is the Caspase enzyme, which is in the apoptosis. This plays like a huge vital role in apoptosis of a cell, and it's actually the intrinsic pathway of apoptosis is the end of the p53 pathway. So you could look at it like that if you want to. But I'm just focusing in on the enzyme itself and the correlation between other enzymes, as there's some order here. So in here we have here's the enzyme, and the active site contains the cysteine, the cysteine amino acid, which I drew out right here. That's your cysteine. Right here, here's the backbone, methyl group, and there's the thiol of the cysteine right there. And what this is going to do is this is going to have a high affinity. There's going to be like a chemical reaction. There's going to be a positive to negative. There's going to be a higher affinity with the cysteine here with a certain sequence of amino acids. It's just the way natural selection set things up. And that's going to be in single amino acid letter codes. The D is aspartate, E is glutamate, and V is valine. Okay? And it's going to cleave at these sites right here. Boom, boom, at both of the aspartate. But you see that it gravitates, positive to negative. It's going to gravitate towards a certain set of amino acids and cleave at certain sites right there. But I just want to, and I'm not comparing apples to oranges. What I am saying is that there is an order, that things just don't go flying around and acting crazy, that there is natural selection has put together, which is an ongoing process, has put together something that has an order to it. So this is protein kinase A catalytic subunit, which is a serine-threonine kinase, which means it's going to add phosphate groups onto either a serine or a threonine on another protein. So, and remember that phosphate group that it adds on there, it doesn't just pull randomly out from the cytoplasm of the cell. It has a site of origin. It's going to take it off an adenosine triphosphate molecule, and then it'll add it to a serine or threonine. But again, it gravitates towards a certain set of amino acids, and that set of amino acids is, it's either arginine, lysine, then X is a variable. That could be any amino acid right there, and then a serine or threonine here. And this is the one that's going to add a phosphate group to. Then down here, lysine, arginine, variable 1, serine, threonine. Again, this is one that'll be phosphorylated. And this will be arginine, variable, serine, threonine. And this is lysine, variable, serine, threonine. Now, if it's any one of these, and you'll see the pattern right here. It involves either arginine and lysine, and then a variable unknown amino acid before the serine or threonine, and I'll add a phosphate group to that. So again, you can see this here is has an affinity to a certain set of amino acids. And it's going to cleave here and cleave here with the caspase, and over here this is going to add a phosphate group, but again, it's a certain set of amino acid sequence there. So things don't just cleave at any one point, and it just doesn't add phosphate groups to wherever it wants to. There's a certain set of order within the chaos due to evolution via natural selection. And that's an ongoing process. So again, quick video, Rich, CNA. See you later.